Thank you for the introduction. I'm Kenji Fujimoto from Hirosaki University. I'm pleased to have this opportunity to speak to you about our cell tracking study. Okay. Cell tracking is a task to track cells in the time-lapse image sequences and extract each cell's moving trajectories. Because cell migration plays an essential role in many processes such as development, immune responses, so the analysis of obtained trajectory is important to understand such processes, especially for the analysis of long-term changes of cell behavior. Tracking each cell for many frames without missing or target switching is crucial. One of the difficulty of cell tracking comes from the cell's appearance. In fluorescence microscopy, cells are visualized as a group of light spots. So a single cell and a group of multiple overlapping cells are very similar and visually indistinguishable. In this case, cell 1 and 2 are getting closer and now overlap. But in these frames, these two cells look like as if they are one single cell. And existing methods are not robust for this situation. Existing methods are mainly based on multiple object tracking methods, but in general object tracking, overlap, also called occlusion, is much easier to detect because each tracking target has different appearances. So existing methods usually fail to track cells when they overlap and produce fragmented trajectories. So to overcome this issue, we propose a novel cell tracking method that associates pre-overlap cells in these frames, and post-reappearance cells in these frames. So it tracks each target while recognizing whether it is a single cell or multiple overlapping cells. So we name our method Overlap Aware Cell Tracker, OACT. So let me explain each step of our algorithm. The first step is just tracking by a regular tracker. Using arbitrary MOT-based tracker, obtain the initial cell trajectories. In this case, this graph represents cell 1 is tracked from frame 1 to 4 and then missed. Then cell 2 is tracked from second frame to 8th frame. And the second step is overlap and reappearance detection. OACT detects each cell's overlap in a very simple nearest neighbor manner. Suppose that cell A is tracked and detected in this frame and then missed. And the other cells B and C are also tracked in the cross location to cell A. And in the next frame, if cell B and C other cell are closely located, then OACT infers that cell A and the nearest one, in this case cell B, are overlapping. And reappearance detection is similarly performed in kind of inverse way. And in this step, the obtained trajectories can be modified like shown here. And now we name the pre-overlap cells as S1, S2, and S3, and post-reappearance cells as D1, D2, and D3. The third step is the estimation of the matches between pre-overlap and post-reappearance cells. For each detected reappearance, OACT associates the pre-overlap and post-reappearance cells. So we now can obtain each image of pre-overlap cells and post-reappearance cells, and then pass to the triplet network and embed in the latent space. Then we can compute a similarity matrix, and now we can obtain the matching. The triplet network used in this process is one of the deep metric learning model, and it embeds given image into the latent space so that same class, in this case, same cell images are closely located and different class images are far apart. The final step is the modification of the trajectories. We now have this trajectory and matching. And in this case, pre-overlap cell S1 and post-reappearance cell D2 are associated. So we now modify the trajectory like this. So extend the cell edge trajectories to the 8th frame and copy the location of this, and these points. And the matching between S2 to D1 and S3 to D3 are similar processed. And we now obtain these three trajectories. And in addition, we propose to apply video frame interpolation for pre-processing. In fluorescence imaging, the frame rate is usually forced to set low to avoid photobleaching or phototoxicity. But this makes each cell's interframe movement larger, and a simple nearest neighbor-based overlap reappearance detection may be difficult. So we apply video frame interpolation in prior to tracking and synthesize the interframe images for the given video and improve the frame rate like shown here. Next, we show our experimental results. We used 13 time-lapse image sequences of leukocytes obtained through two-photon microscopy. We compared our method with Tractor. It is one of the well-known CNN-based MOT method. And here's the result. In terms of false negative, it is a number of missed cells and number of cell ID switches and number of trajectory fragmentation. OACT outperformed Tractor. So the ability to continuously track each cell across many frames was improved by OACT. In contrast, in terms of false positives, which is the number of false detections, was outperformed by Tractor. We consider the cause was maybe errors in overlap reappearance detection or match estimation. This slide shows the example results. In this case, two cells are getting closer and overlap. Existing method misses one of the cell because it recognizes this is one single cell. But our method successfully recognizes this is a group of two overlapping cells and continuously tracks from both of the cells from the pre-overlap frame to the post-reappearance frame. Next, we show the two ablation study results. The first version is without matching, so just perform interpolation before tracking. And second is without interpolation, so without pre-processing. These two versions, in terms of false negatives, ID switches, and fragmentation, outperformed Tractor, but did not outperform the full version of OACT. So without matching, overlapping cells could not be accurately tracked, and without interpolation, pre-processing, errors in overlap reappearance detection occurred more frequently and resulted in poor tracking performance. This is the conclusion. We proposed a novel cell tracking method, OACT, and outperformed existing tracking methods in terms of continuous tracking. We consider this ability contributes to the analysis of long-term behavioral changes in cell migration. As a future work, we are considering to develop a method to distinguish between overlapping cell and the disappearing cell. Also, we are considering to use appearance history or motion features for more accurate estimation of matches. That's all. Thank you.
Thank you very much. And are there any questions? Yeah, please come forward. Yeah, thank you.
So, have you ever tried out to use your algorithms to detect different cell types? Because different cell morphology could be different results, right? Have you ever used your algorithm to apply to different cell types?
You mean different data?
Just as a fibroblasts, right? Yeah, was rounded cell type. Have you ever tried out to apply your method to different cell types?
Actually, currently no, but it's a future work.
Okay, good. Thank you.
Yeah. So Kenji, I have a question. Can you comment on the computational efficiency of your model training? I mean, how long do you develop a much high-performing, accurate model?
You mean the computational
time?
time?
Yeah.
Actually, we haven't conducted precise analysis, but it requires high performance, high computational complexity because it considers the matching between two groups of cells for each reappearance of time. So in current status, it is not for the real-time application for tracking the real time and do something using the results.
So your model does not require the annotation by the experts, like it can be automatically trained.
Actually, annotation is required because
Still required the segmentation, the kind of manual curation process involved.
Actually, manual segmentation is not required, but manual tracking is required because the triplet network used to measure the similarity between the group of cells trained with using the information this cell and this cell are same cell and these images are different.
Yeah.
So, and currently we are using the tracking of annotation for making a training data.
Yeah, we still have time for a few more questions. Is there any other question from the audience? Yeah.
I'm not sure that how can you distinguish between the the cells between D2 and D3?
I think it's a hard question, but because we are using the triplet network to measure the difference of a deep learning model to measure the similarity of two images of a cell. But I think the small difference of color distribution or shape distribution is the point of difference. I think so.
So your model can learn the shape or color of these cells?
Yeah, actually, currently I'm using a very simple CNN model, but yeah, using more complex or more sophisticated model, we can consider you can consider more complex features.
Thank you.